Good morning everyone. I am Ashik and today I will be presenting on microbial degradation of environmental pollutants with a focus on industrial solvents. So industrial solvents are essential in modern industry but they also pose a major environmental challenge. In this talk, I will explain what industrial solvents are, why they are pollutants and how microorganisms can help degrade and detoxify them in an eco-friendly way. So industrial solvents are chemical substances, mostly liquids, used to dissolve, extract, or carry material without altering them chemically. They are used everywhere in paints, coatings, adhesive, pharmaceuticals, cleaning products, and manufacturing. However, due to improper disposal, spills and leaks, these solvents often contaminate the air, soil, and groundwater. Even small quantities can cause long-term pollution because many of these compounds are toxic, stable, and persistent in the environment. So while they are vitally important, they have become one of the most common class of chemical pollutants worldwide. So let's look at why these solvents are such a serious environmental concern. At first, toxicity. Many solvents are poisonous to humans and animals. For example, benzene is a well-known carcinogen, and others can damage the nervous system, liver, and kidney, etc. Second comes the persistence. Some solvents, especially chlorinated hydrocarbons like trichloroethylene, resist natural breakdown and remain in soil and groundwater for many years. And the third will be bioaccumulation. These compounds can so industrial solvents can accumulate in fatty tissues of microorganisms, they move up the food chain from plankton to fish, then from fish to humans. Even at low concentration, they can lead to long-term exposure and health risks. All of this highlights the need for sustainable and effective degradation strategies, especially using microorganisms. So the need for microbial degradation. Conventional methods like these physical methods, chemical methods, thermal treatments, etc. are expensive and often just transfer pollutants from one medium to another. As I've given here, they transfer pollutants from one medium to another, for example, from water to air without truly destroying them. In contrast, microbial degradation is eco-friendly because microbes convert toxic solvents into harmless products like carbon dioxide, water, and biomass. Then they are cost effective. That means with low operational and maintenance requirements. Then comes the self-sustaining since microbes can adapt and reproduce at contaminated sites. Then versatile, that means the capability of degrading a wide range of compounds. Microbial methods not only remove solvents but also restore ecosystem balance. Thereby protecting water quality and prevent bioaccumulation in food chains. Here I have given some environmental and health benefits that include reduces toxic load in ecosystems, prevent bioaccumulation in food chains as I have mentioned, then protects drinking water source from solvent contamination, minimizes greenhouse gas emissions, etc. So common industrial solvents and their impacts. Now let's go through some common industrial solvents and some environmental impacts. Some of them are, like example, chlorinated solvents like trichloroethylene and perchloroethylene are used in degreasing and dry cleaning. They are toxic, carcinogenic, and highly poison, often contaminating the groundwater. Next group comes the aromatic hydrocarbons such as benzene, toluene, and xylene are common in fuels, paints, plastics, etc. They often release volatile organic compounds contributing to air pollution and smoke formation. Benzene, as I mentioned before, is a carcinogen. Then the third group, that the alcohol, ketones, and phenols like ethanol, acetone, and phenol are also used in disinfectants, in coatings. Alcohols degrade relatively easy, but phenol is toxic and can persist in wastewater. these pollutants reach soil, air, and water, causing serious ecological and health issues that require biological treatment approaches. So several different groups of microorganisms are involved in solvent degradation. We can look at some of the groups. First comes the bacteria. Bacteria such as Pseudomonas, Phytida, and Rhodozoa species are primary degraders of hydrocarbons or chlorinated hydrocarbons and chlorinated solvents. Dihalococoids McCarthy performs anaerobic dechlorination of TCE and PCE. That means trichloroethylene and PCE. Fungi. Next group is fungi, also known as oxidative degraders. Fungi, particularly this white rot fungus, that Phanerochaete chrysosporium, degrades phenols and chlorinated solvents using oxidative enzymes such as lignin peroxidases. Then the third group, RK. RK contributes in extreme or oxygen deprived environments. The methanotropic and halophilic RK can oxidize short chain hydrocarbons. And the fourth group, the microbial consortia or cooperative degraders. Microbial consortia, or we'll say as mixed communities, that several different type of microorganism are often they are most often most effective when combined. For example, when one microbe transforms a solvent into an intermediate, and the other microbe can complete the process. This microbial diversity ensures complete mineralization of even complex solvent mixtures. Now we look into the mechanism of microbial degradation. The mechanism of microbial degradation follows mainly two main pathways. One is aerobic pathway and the second one is anaerobic pathway. In aerobic degradation, aerobic pathway, it occurs only in the oxygen availability. Microbes use oxygen to oxidize and hydroxylate the solvent molecules. That is, the oxidation or hydroxylation will happen, and also ring cleavage for aromatic amino acids will happen. For example, Pseudomonas Phytida converts toluene into catechol, which when enters the TC cycle, ultimately forming carbon dioxide and water. So you can see the end products of aerobic pathway are carbon dioxide, water, and microbial biomass. Then comes the anaerobic pathway. This takes place in oxygen depleted conditions, such as in groundwater, deep soil contamination, etc. Here, microbes use reductive dehalogenation and methanogenesis, sulfate or nitrate reduction, etc. As an example, dihalococoides McCarthy dechlorinate TCE into an harmless ethene. Key enzymes as I mentioned below on the bottom part, key enzymes involved in this degradation include monoxygenases, dioxygenases, dehalogenases, and peroxidases, all of which catalyzes these biochemical transformations. So, if the factors affecting biodegradation, there are four different factors that affect the biodegradation of industrial solvent, namely physiochemical properties of the solvents, environmental condition, nutrient availability, and presence of co-substrates. Physiochemical properties of the solvent include hydrophobicity. That means how the solvent interact with the water molecule. If it is hydrophobic, it won't mix with water, and thereby reducing the ability of the microbes to degrade that solvent. The second one comes the volatility. Then if it get mixed into the air all of a sudden without giving time for the microbes to act on them. Then solvent toxicity. Some solvents may be toxic to the microbes itself, and then solubility in water, etc. And environmental conditions such as pH, temperature, oxygen, and moisture must be optimal for microbial growth. That means different organisms for different solvents need different conditions. Then comes the nutrient availability. Microbes need nitrogen, phosphorus, sulfur, and other trace minerals for growth. Lack of nutrients will slow degradation even if the solvents are present. we can add nutrients that can accelerate degradation rates. This process is known as biostimulation. Then the fourth one are the presence of co-substrates or known as co-metabolism. Uh, Here some solvents degrade only when other carbon salts is present. For example, methanotrophs degrade TCE only while oxidizing methane. So bioremediation approaches for solvent degradation. So bioremediation strategies are mainly classified into two, namely in situ methods and ex situ methods. In situ methods are the technique of bioremediation in which bioremediation is done in the site of pollutant itself, and in ex situ methods the bioremediation is often done in the solvents are transported to a particular setup and then they are degraded. So in situ methods include biowinding, bioaugmentation, natural attenuation, etc. This biowinding is the process of supplying air and nutrients to contaminated soil to enhance aerobic degradation. That's it. Then comes the bioaugmentation. Bioaugmentation is the method of introducing specialized microbes when native species lack degrading ability. That means when the microbes that is already present lack the ability to degrade the solvents, microbes that are able to degrade the solvents are brought in, and thereby increasing the ability to degrade. Then comes the natural attenuation. That means relying on existential microbial activity and natural process to reduce pollutant level. That means just maintaining the waste there and waiting for the nature to do its role. And secondly, it comes the ex situ methods, which involves bioreactors, composting, and slurry phase treatment. In bioreactors, treating contaminated materials under control, contaminated water or soil is treated in controlled, that is optimized pH temperature and aeration. First, optimized pH temperature and aeration for faster solvent degradation. For example, treatment of phenolic wastewater in aerobic bioreactors. Then comes composting. Composting is a mixing contaminated soil with organic amendments such as manure, straw, etc. It generate heat, oxygen, and microbial diversity. These things accelerate the degradation, accelerate the breakdown. Composting is effective for semi-volatile solvents like phenols. Then next comes the slurry phase treatment. This soil is mixed with water and treated in slurry bioreactors. It provides high contact between microbes and solvents, common for chlorinated and aromatic solvents. Each approach is selected based on the contamination type and its characteristic. That is in situ, it can either be in situ or ex situ. It's based on the contamination type and the site characteristic, either in situ or ex situ is selected. So challenges and limitations in microbial degradation of industrial solvents. Despite its promise, the microbial degradation faces, of course, several challenges, including solvent toxicity. That means these microbial solvents can inhibit microbial growth or enzyme function. Second comes the slow degradation rates for allergens. That especially for allergenated solvents, this slow this can lead to slow degradation of allergen solvents, can lead to accumulation of toxic intermediates like vinyl chloride. Then comes the limited bioavailability. Some solvents bind strongly. Bind strongly. Some solvents bind strongly to soil particles or evaporates quickly, reducing microbial access. Then the third one, they have the environmental variability. Fluctuations in pH, oxygens, and nutrients, etc. can reduce efficiency. That means depending on the environment, the temperature, pH, etc., the rate of degradation can either increase or decrease. Then mixed contaminant complexity. That means several heavy metals, organic, etc. can become mixed in the environment and and can reduce efficiency of this degradation. Uh, when the organism is trying to degrade one solvent, the other may act toxic to the organism, thereby complicating the degradation pathways. So we'll now look into the recent advances that has brought major innovations in the field of microbial degradation. The first one, is the sorry for the inconvenience. The first one is microbial consortia and synthetic biology. That means consortia based remediation and synthetic biology approaches. Consortia, as I mentioned before, is the use of mixed population of different microorganisms, thereby increasing the efficiency of this microbial degradation. Synthetic biology approaches means the use of recombinant organisms in which extra degradative pathways are involved in it. Then comes the nanotechnology assisted bioremediations. Nanoparticles like zero-valent titanium dioxide enhance microbial activity and catalyzes solvent breakdowns. In nanotechnology assisted bioremediation, nanoparticles can be used as catalysts or supports. Then nanoparticles, microbe hybrid, then also the controlled delivery systems. Then comes the omics-based approaches such as metagenomics, transcriptomics, proteomics, etc. These allow scientists to design precise and omics-based approaches such as metagenomics, etc. allows the scientists to design precise bioremediation. That means by using these techniques, that metagenomics, that study of the whole genes, then metatranscriptomics, study of proteins, etc., it will allow the scientists to design precise bioremediation strategies. To conclude all of this, why microbial degradation is better than other conventional methods? So microbial degradation of industrial solvents offers a sustainable and eco-friendly alternative to conventional remediation techniques. Unlike physical or chemical methods that are often costly and generate secondary waste, microbial processes harness the natural metabolic capacity of microbes to detoxify pollutants. However, complete remediation may require integrating microbial approaches with physical and chemical methods for greater efficiency. Looking ahead, advances such as engineered microbes with enhanced catabolic pathway, targeted biostimulation of native microbial communities, and multi-omics guided strategies hold great promises in developing faster, more reliable, and scalable solvent for solvent pollution. These are the references. Thank you.